all of you. Ladies and gentlemen, Albert Einstein once said that every human being is bestowed with just enough intelligence to be able to see clearly how utterly inadequate that intelligence is when confronted with what exists. Which is why we as intellectuals get together and engage in research to get to know what we do not know. And we are also gathered here today for that reason. We warmly welcome you to the International Research Symposium of Rajarata University 2021.
සුබෝදෑසනක් ඔබ සැමට වඩාත් සුන්දර යහපත් ලෝකයක් බිහි කිරීමට මිනිසා විසින් මිනිසා උදෙසා සිදු කරන නව අත්හදා බැලීම් නව සොයා ගැනීම නව පරිශ්‍රණ ඉවහල් වෙනවා. එවන් වූ කැපකිරීම කරන මිනිසුන් උදෙසා වේදිකා තනන්නේද මිනිසුන්මයි. ඒ වගකීම මනවින් අවබෝධ කරගත් ශ්‍රී ලංකා රජරට විශ්වවිද්‍යාලීය තාක්ෂණ පීඨය මගින් මෙවර සංවිධානය කරනු ලබන 2021 අන්තර්ජාතික පරීක්ෂණ සමුළුව සඳහා සැපැමිණි ඔබ සැම ඉතාමත් ගෞරවයෙන් ආදරයෙන් යුතුව පිළිගනු ලබනවා.
To commence the inauguration ceremony auspiciously by keeping in line with the rich traditions in Sri Lanka, let us now join in lighting the traditional oil lamp. We would like to invite the following dignitaries to join us in this customary tradition. First and foremost, Professor Sanjeevani Ginigaddara, the Vice Chancellor of Rajarata University of Sri Lanka, followed by Professor Buddhika Jayatilaka, the Dean of the Hosting Faculty, Faculty of Technology, Dr. UGAI Sirisena, representing the Dean of the Faculty of Agriculture, Professor EMKRB Edrisingha, the Dean, Faculty of Applied Sciences, Professor W.P. Vijayawardena, the Dean, Faculty of Management Studies, Professor Sunith Agampudi, the Acting Dean, Faculty of Medicine and Allied Sciences, Professor Tusitha Mendis, the Dean, Faculty of Social Sciences and Humanities.
Followed by Dr. Chaturanga Bamunuarachige, Chairperson ISymRU 2021, Dr. Pubudu Kumarage, Coordinator ISymRU, Dr. Mithila Bandara, Secretary ISymRU, Ms. A.S. Sirivardana, the Librarian, Mr. N.C.S. Senaka, the Acting Registrar, and Mr. D.D. Upananda, the Bursar.
May we please grace to the traditional oil lamp. Thank you. May we please rise to the university anthem.
ප්‍රඥාලෝකය දැල්වූ ජය සංකේතය මිහින්තලේ වරදේ වරදේ ශ්‍රී ලංකා රජරට විශ්වවිද්‍යාලය සැරදේ ප්‍රඥාලෝකය දැල්වූ ජය සංකේතය මිහින්තලේ වරදේ වරදේ ශ්‍රී ලංකා රජරට විශ්වවිද්‍යාලය සැරදේ උරුමය සැකසිය හැදියාවයි මතු කළ සම්මා දර්ශනයයි ශිල්ප ශාස්ත්‍රනිකේතනයයි බුද්ධිය ශක්තිය ප්‍රබෝධයයි ප්‍රඥාලෝකය දැල්වූ ජය සංකේතය මිහින්තලේ වරදේ වරදේ ශ්‍රී ලංකා රජරට විශ්වවිද්‍යාලය සැරදේ දැනුමනුවන දේශය වෙනුවෙන් නංවමු සුජීවනය වඩමින් විද්‍යාඥාන ගවේෂණයයි මානවවාදී විහරණයයි ප්‍රඥාලෝකය දැල්වූ ජය සංකේතය මිහින්තලේ වරදේ වරදේ ශ්‍රී ලංකා රජරට විශ්වවිද්‍යාලය සැරදේ
To welcome all of you to this occasion, I would like to invite Dr. Pubudu Kumarage, the coordinator of ISymRU 2021, to welcome all of you by his official welcome address.
සුබෝදෑසනක් සියලුම දෙනාටම. Good morning to you all. වැඩම වදාළ ශාස්ත්‍රපති පණ්ඩිත ශ්‍රී ලංකා රජරට විශ්වවිද්‍යාලයේ කුලපති අවසරයි ඊතල් වැට්නුවේ ඥානතිලක අපේ ස්වාමීන්ද්‍රයන් වහන්සේ ශ්‍රී ලංකා රජරට විශ්වවිද්‍යාලයේ ජ්‍යෙෂ්ඨ කතිකාචාර්ය අවසරයි ඕලන්දවත්තේ චන්ද්‍රසිරි ස්වාමීන්ද්‍රයන් වහන්සේ newly appointed Vice Chancellor of Rajarata University, Professor Sanjeevani Ginigaddara, all the deans of the faculties and heads of the departments, directors of the units, admin officers of the Rajarata University, all the fellow staff members of Faculty of Technology and non-academic staff, and all the students. It is with great pleasure to welcome you all to the International Symposium of Rajarata University, ISymRU 2021. ISymRU 2021 has become a great platform to share and disseminate the knowledge gathered and created by the scientists in Sri Lanka and also by the scientists throughout the world. We have this symposium throughout 2 days, today and tomorrow. Not only to this ceremony, I welcome you all to this symposium to join with the sessions and get the knowledge from the works of those scientists. Behind all this glitter and glamour, actually there's a lot of hard work. That's why I invite you all to get the best out of it and to share the knowledge. And with that, without taking any more time, I would like to welcome you all again to this symposium, ISymRU 2021. Thank you very much.
Thank you very much, sir. The next item in the agenda is the keynote speech. I would like to invite Dr. Dinum Pereira to introduce our keynote speaker and to coordinate the session.
For decades before COVID-19, scientists warned that a pandemic was inevitable and urged preparations. But unfortunately, governments around the world did very little. When it arrived, COVID-19 laid bare the fragility of many of our systems, including global food supply chains. You know what? A plant pandemic could be the next. Across farms worldwide, there are now warning signs of a pandemic in food crops. Therefore, the agriculture industry does not have to make the same mistake, and it's high time we greatly focus on plant health. Our first keynote speaker is one of the leading plant biologists in the United States, and his lab primarily focuses on molecular genetic and biochemical processes associated with interaction between plants and pathogens. He served as the foundation professor of plant, soil, and microbial sciences at Michigan State University. Lately, he joined University of Tennessee, Knoxville, as Associate Vice Chancellor for Research and Innovative Initiatives. Here to share his take on the impact of environment and pathogens on plant health and agricultural ecosystems. Please welcome Professor Brad Day.
Okay, thank you, Dinum. Can everyone see my first slide okay? Okay, let's get started. So I appreciate the invitation to join all of my friends and colleagues back at Rajarata University during this difficult time. It's been great knowing that you guys are doing well and still communicating with all of us on research objectives that we initiated about 3 years ago. So this afternoon, or excuse me, this morning or this evening, depending on which part of the planet you're located on, I'm going to talk a little bit about the research in my lab and also some of the research that we're doing collaboratively with Rajarata University. And so I've described this talk as kind of the perfect storm. And as Dinum alluded to, we may in fact see the perfect storm one of these days. Hopefully not, but to be prepared, a lot of research in this area is focused on the convergence of multiple stress events in plants. And so what I'm going to talk about today are two of those primary events, and that is the threat that pathogens impose on plant systems, as well as the abiotic threat that the environment and changing environments impose on plant and agricultural systems. And so, as Dinum described, research in my lab is primarily focused on the interactions between plants and pathogens. We are a collection of geneticists, cell biologists, biochemists, and a lot of bioinformatics and genomics research are undertaken in the lab to understand the precise mechanisms that plants utilize to defend against an array of biotic and abiotic threats. And what I'm going to talk about today is really the convergence of these processes, as well as the development of translational tools and solutions for sustainable agriculture. And the latter part of my talk is going to primarily be focused on the collaborative research that we're doing under the guise of the Asian Development Bank with Rajarata University. And so then just to get you kind of a preview into some of more of the translational applications that my lab is interested in, is our technology is poised or positioned at the intersection of food and environment and sustainability. And so we're very interested in how plants evolve to become resilient in the face of numerous stressors. And we use technology in a variety of ways, shapes, and forms to address these challenges, whether it's state-of-the-art point of contact technologies to diagnose diseases, or whether it's in the lab to dive deeper into the genetic makeup of plants and pathogens to understand how they're communicating, how they're talking to each other. And this has broad applications, not only to protection of plants against pathogens, but also against changing environments, rapidly changing environments, the threat of drought, the threat of pandemics or epidemics, and diagnostics, food production, so food safety, food security, and by translation, human health. The solutions that we're working towards are rapid, translatable, and again, kind of point of contact. We're very interested in cost-effective measures that are accessible to all walks of life, to all farms, big and small, to all technologies, to all partners and countries around the world. And our platform is really an empowering technology. And I'm going to show you a few pictures in the latter part of my slide where students at Rajarata University can really break down the technology and understand the mechanisms that drive this. So what I'm going to be talking about today is not just a plant biologist perspective on how plants and their environments interact, but really how computer scientists view this, how engineers view this, chemists, physicists, et cetera. And so with this, there are numerous challenges out there, but there's a lot of technology, a lot of innovation taking place. I'm sure many of you are familiar with the marvelous work that USAID is doing, and by translation or by similarity, the Asian Development Bank is funding fundamental research that can have translational or intended outcomes. There's gene modification technologies, CRISPR-Cas, genetically modified organisms, whether to produce these organisms or to not produce these organisms. That's still largely a point of debate. And there's also new technologies for imaging, for capturing data. But at the root of all of this is, what do we do with this data once we have it? And that's one of the partnerships that we have with Rajarata University, is understanding how to use this massive amount of information for the betterment of food production systems to sustain resilient and durable solutions for human health. And so largely my lab has primarily been focused on plant environment interactions. And these are coming really two forms. There's abiotic stress, which are, events such as heat, drought, cold, flooding, et cetera, or biotic stress. And this is where a lot of my lab has been focused for the past 15 years. And that's understanding how pathogens attack plants, why most plants are resistant to most pathogens. Yet it are those few occasions when a pathogen is successful in occupying its host, infecting its host, and causing disease that we really need to be concerned about. And so if we think about what's going on in the environment, plants are sessile organisms. They don't get up and move around when they encounter danger. And so to defend against this myriad of threats, they're really constantly surveying their environments. They're surveying for changes in temperature, changes in water availability, changes induced by virus infection, insect chewing, bacterial infection, bacteria inoculation. And on the left side, what we see is that beneficial microbes, such as those that provide enhanced growth or enhanced features or properties to the plant, they really converge on those events that pathogens also initiate. And so how do plants recognize friend from foe? And so this is one of the key areas that research in my lab has focused on. But on top of this, many of these friend or foe recognition events are also triggered by rapid changes in the environment. And whether these are long-term sustainable and ultimately detrimental changes, or if these are rapid transient changes, plants have evolved the innate ability to respond to their environment through a variety of processes. And so they respond to these changes with numerous physiological and genetic changes. Many of these are very rapid. So if you can think about your own interactions with your environment, when you walk outside, you can very quickly tell if it's hot or if it's cold. You can very quickly be alerted if an insect has stung you, or if you've injured yourself and the insect or an animal has broken your skin. And all of these events occur very rapidly, and they initiate intracellular signaling, processes that require changes in the cell dynamics, changes in your host physiology to prevent further injury, infection, and or disease. And so these are just some of the events that my lab has been primarily focused on and identifying throughout my time at Michigan State University. And all of these events in plants are also highly conserved in us as humans. And so kind of the first takeaway point I want to make at this time in my presentation is that plants respond to their environment much the same way humans do. Yet plants have evolved the ability as organisms that can't move around. They don't have sophisticated immune systems, yet they can respond to a myriad of threats, symbiotic and abiotic interactions. So how do plants do this? So in the case of us humans, if we are being challenged or attacked by an insect, we can simply swat that away, walk away, and move to a safer environment. But how do plants do this? They reside in a single environment throughout their life cycle, reproduce through seed production, and many of those seeds fall to an area that is adjacent to the plant. Some can be carried off further by animals, but plants are really rooted in their environment. So how do they grow and survive at the same time when all of these threats? And so this is one of the questions that my lab is very interested in. And we have two papers in process now that are focused on a single gene that when we remove this gene from the plant, plants are more susceptible to drought, they're more susceptible to disease, they're more susceptible to elevated temperatures, ultimately will die without this gene, and they have this small stature as a function of this gene. But where our research has taken us is that if we can change the expression of this gene, we now have plants that are able to sustain drought, able to resist pathogen infection, able to withstand elevated temperatures, and do not die in the presence of prolonged pathogen infection. And so this leads us kind of to the first technology that we're using in the lab, and that is to identify the genetic makeup and really kind of the genetic convergence of where this gene lies within the plant immune system. And so this is where really kind of just summarizing recent studies by two postdocs in the lab, one ISymRU Liu, who's now a assistant professor in Taiwan, and a Sri Lankan, Sarupa Samaradivakara, who some of you may know. And what Sarupa and ISymRU have demonstrated is that the expression of this gene, the presence of this gene, when upregulated, can impart numerous benefits to the plant, both in terms of biotic stress challenges as well as abiotic stress challenges. And what we found through analyzing this study was that many of the processes, or excuse me, many of the genes converge on these similar processes that I described to you earlier, such as cold, heat, toxin, drought, immunity or pathogen infection, and other abiotic conditions such as elevated salt. And so what we're learning about this is as plants rapidly perceive their environment, or excuse me, as plants perceive their environment, they're able to rapidly induce changes in their physiology to adapt to that environment and to successfully defend themselves in that harsher environment so that they can continue their growth, reproduce, make seeds, and continue their life in those environments. Now I'm going to make a really rapid transition into a new technology because I don't want to spend a lot of time going through an introduction. I've given this talk, or at least portion of this talk, to some of you in the past, but where my lab is really interested in is how can we use lab-based technologies to understand how we can prevent or how we could diagnose plants in a field-based environment. And so this work started out about 4 years ago by a graduate student, Amy Bateson-Young, and has now since been continued by Sarupa. And what we're able to do is develop a lab-based diagnostic assay that is portable, that we can take to the field and understand how plants are behaving and responding to their environment. And what we did is we're able to get this assay to work at a very high level of sensitivity. So we can take this test into the field, understand what types of threats may be imposed on the plants or this crop or an entire field, if you will, and at a very sensitive, rapid pace, understand what types of threats we're dealing with. So how do we mobilize lab-based technology to the field and mobilize it in a way, package it in a way that is accessible to a broad range of users? And these users can include farmers, they can include professors, students, people that grow and process food, people that trade food, import, export. So this application that I'm going to describe to you in the next 5 or 6 slides has utility across a breadth of food-based and agricultural applications. And so the way we've organized a lot of this research, and many of you in the audience may have met my colleague David Kramer, who's also a professor at Michigan State University. And Dave developed this ingenious handheld device. And the really cool thing, I've got my prop here ready. I don't have my device with me, but I have a very common item here you all recognize as stapler. This device is approximately the size of a stapler, a little bit lighter than a stapler, but the device possesses a number of key attributes that enable us to take lab-based methods, lab-based technologies, and move them into the field, collect data, collect information, send that back to the lab, back to the cloud, back to collaborators all around the world, where we can rapidly analyze it and then push solutions back to the field. And so to do this, we can create projects, select sensors, add some methods, analyze the results, and then the most important part, as Denham mentioned in her introduction, as academics, as scientists, as policymakers, as government officials, as deans and directors and chancellors, we can discuss the results. We can come up with viable solutions for the problem at hand. And this is the next take home that I really want to say, express to the students, is that the discussion around science needs to continue. It's really important, regardless of your location on this planet, regardless of where you are, what country you're in, what level you're at, whether you're a student, professor, we all need to get together and talk more about science. I am wildly and 100% convinced if we had discussed and been prepared and communicated this pandemic much better and much faster, the pandemic would be over and I would be sitting in the audience with you today. And so the idea for this is a field-based marketplace where people can gather information, share this information. And this is just some of the highlights from a publication from Dave's lab a number of years ago, where each one of these dots represents a single data collection event. And in just a matter of hours, they were able to collect hundreds of data points describing the conditions, the environmental and plant health conditions in this field. You can map this out to very high resolution, and now they're working on a machine learning based process that we can now go into the field, and it helps guide us. So as we are collecting information, Dave is developing the technology that a smartphone is actually much smarter than the typical smartphone, and it will actually guide us into taking measurements and tell us where to take these measurements. And so one of the key features I think that we've really moved forward in is portability. And so as we were developing these projects, as Sarupa was developing these projects, we started looking at the available technologies that we're relying on in the lab and said, this is much more sensitive. This is a technology we need to provide more data, provide more information and more rapid information. And so Dave's lab developed a handheld portable device that's equivalent to this device over here, which is in the neighborhood of $30,000. He's developed and built a multi-spec plate reader for about $75 US. And what this has allowed us to do is to move forward with this project that's funded by the US Department of Agriculture. And now a portion of this project is also recommended for funding and funded by the Asian Development Bank in partnership with Rajarata University. And Chaturanga and Denham are a very important part of this collaboration. And what we've been able to do is to start map out how we're going to tackle some of the greatest challenges, I think, that are facing agriculture, food safety, food security, and human health in Sri Lanka. And our test study is going to be first looking at rice. And so again, back to this idea that we have, we're going to develop this point of contact diagnostic tool, but also to have lab-based experiments where we're going to be understanding the mechanisms that plants utilize to protect themselves. But as we move into the field, we can identify these events very rapidly and provide solutions. So how do we get there? And so what my lab was very interested in was a process of nanotechnology. So this is a nanobiosensor-based technology. And I'd like to give some common examples here that are very popular around the world. Golf balls, advanced tools, tennis balls, for example, a pregnancy test, a glucose meter. These are all nano-based technologies that we're all very familiar with. And so what we did was we wanted to make a technology and an application that people are familiar with. They understand nanotechnology. They can understand how our device works. It's a basic colorimetric device that can be used for the development assays against pathogens for plant breeding. So Sarupa has tested this and is working with colleagues in Turkey to understand how this might be used to detect the presence or absence of genetically modified DNA elements in plants, also for pathogens. In Sri Lanka, we're very interested in how heavy metal contamination, arsenic, for example, can be detected and present in either plants, soils, or water, and then expanding these into more toxin-based applications or pharmaceuticals. So any of these substrates really now become a nice analysis for us to feed into to begin to detect at point of contact in the environment, in a field, in a stream, in an agriculture setting or a natural landscape, communicate that data back to a group of individuals, scientists, academics, et cetera, and understand what we're dealing with. So that really describes, I think, this pull-push technology. We want to pull information from the field and push a solution back to the field. So as I mentioned, we're working with scientists and students and farmers. Import-export is one of the opportunities I think that we haven't really tackled, but I think it makes a lot of sense. A lot of food is moving across borders these days. A lot of infected individuals are moving across borders these days. And if we can make one small step by understanding, is our food safe? Is it free from toxins or bacteria or elevated levels of hormones or pharmaceuticals? I think ultimately that is a driver for food safety, and that protects all humankind. And so just to give you kind of some highlights of the things that we're doing with Rajarata University, and this was one of the best trips I've had to Sri Lanka. I've had 4 trips, and this certainly is actually the best trip I've had. And this, I believe, was in 2019. My years are running together these days. Apologies. 2019, and we visited a smallholder farmer, and we were able to demonstrate our technology to the smallholder farmer and diagnose some of the citrus plants that they were having. And this is an example of where you don't have to be a plant biologist to understand and utilize this technology. And so these are some of the students from Rajarata University. And what they're doing is they're breaking down the software. They're understanding how this process works. And so this was really exciting to see all of these students on the first day, just crowded in around the computer, understanding how to develop and enhance the value of this technology. And so one of my big dreams, I'm obviously now moving on into more administrative positions at the University of Tennessee, but one of my big dreams still is, how do we move information? How do we share information? How do we transform the technologies that we use to capture, send, receive, and diagnose information? And so one of the ideas that I'm still working on somewhat adjacently to my adjunct position at Michigan State University is with a group of about 25 professors, students, and faculty and staff at Michigan State. And the idea is to develop bioengineered system for environmental monitoring. And so this is a grand project where we utilize the innate ability of plants to detect and sense their environment and to communicate these signals as a function of novel sensors, and to allow these sensors to be picked up by man-made engineered systems, and to send these great distances where they can be transformed, translated, understood, and reacted to. And so this is kind of the dream. This would be the 2030 or the 2040 plant of the future, where you could have these plants growing in a field, and they would be the sentinels. They would be out there surveying the field, monitoring for all of the threats that the human based technologies cannot sense. So these plants could be sensing changes in soil behavior, changes in microbial composition, changes in temperature, changes in insects, changes in volatiles that could be toxic to humans. Plants have a very sensitive way of detecting these. So this, again, just keep this in mind. This is something that I think we can utilize and grow our collaboration with Rajarata University by understanding how chemists think about food security and plant biology, about how engineers and computer scientists and even business people, how we all think about the one thing we have most in common, and that's food. And so with that, I'd just like to thank the great collaborators, the scientist, Sarupa Samaradevakara, for her work in both projects, the NDR1 related project focused on abiotic stress tolerance, elevated temperature, as well as her continuing work in developing new technologies for advanced sensors. And of course, Chaturanga and Denham for providing us with an opportunity to connect to some of the smartest, most ambitious and hardworking students I've had the opportunity of meeting. That trip to Sri Lanka and visiting Rajarata University was really a life-changing experience. And I look forward to the opportunity of returning back to see all of our colleagues and many other colleagues that have helped us along the way, especially Dave Kramer and Atsuko Kanazawa, who developed and are still continuing to grow and evolve the multi-spec and photosync technologies. And so with that, I will be happy to stop and take any questions that you may have. Thank you.
Thank you so much, Brad. What an insightful presentation. It really provided a deep understanding into how biotic and abiotic factors affect plant growth and development, and it's really cool to see like how we can use all those multi-spec to get all those plant parameters and all. So any questions for Dr. Brad?
And so let me apologize. I just saw the messages that my presentation was in presenter mode, so I apologize for that. And Denham, if people are interested, I can provide you with a copy of my slides, and you can share those if people have specific questions.
Thank you, Professor Brad, for joining with us today. It was a really nice presentation, and thank you very much for sharing your research work with us. And I believe all of us enjoyed your presentation. You know, Sri Lanka is a tropical country, and where its economy is vastly dependent on the agriculture sector. And I believe most of your research work can be applied in here. But since you are using high technologies and a lot of high technologies, I'm worried how could we possibly use your technologies in here. Do you have any suggestions on that regard? And if that's so, it will be grateful. We will be grateful for that.
Right. That's a great question. And first and foremost, I'll give you the ultimate answer is that we develop these technologies with countries and partnerships like Sri Lanka in mind, like India and Indonesia and Southeast Asia and Africa. So the technology was first launched in partnership with the Gates Foundation with Dave Kramer's lab through funding into Africa. And so this technology is one that crosses not only geographical borders, but financial borders as well. It also crosses access to technology borders. And so a couple of things I want to point out, and I didn't mention this. So I gave some of the similarities between various types of nanotechnology. And I think everyone's probably familiar with a blood glucose test or a blood glucose monitor, maybe even a pregnancy test. Both of these are based on popular nanotechnology devices. And in those applications, which are vital, those applications cost about $8 to use. And so as we were developing our test, we kept that in mind. $8 is sort of a spot that a typical price for a diagnostic assay would land. For $8, you can do 32,000 assays using our technology. And so if you think about $8, so $8 in the United States is the price of a typical potato virus Y assay that a farmer might use and go into their field to determine if they have a viral infection in their potato plants. For $8, you can test every single plant in the field using this technology. The second part, the reason I showed the picture of the students at Rajarata, was that I'm a firm believer that, obviously DNA is DNA, genes are genes, regardless of where they're from, and computer scientists can basically crack any code that's out there. And as I showed you in that picture, within just several hours of us initiating our last workshop in 2019, these students were crowded around the computer, cracking the code, developing, understanding how this technology works, and able to start thinking about how they might reprogram this device to do specific assays. So I'm not worried about countries like Sri Lanka, particularly countries like Sri Lanka, in your ability to access these technologies. Computer science is certainly, I think, a rising area, a discipline in your country. Engineering and biology are not far behind. And so I have nothing but the absolute confidence that your students in that generation are going to lead in the development, the evolution of these types of existing technologies. So in that regard, I think this is a perfectly accessible example of an advanced technology for food security.
Yeah, thank you very much, Professor Brad. Thank you very much, Dr. Dim.
Thank you, Brad. So, some more questions for Brad?
Thank you, Professor Brad, for your interesting presentation. And I'm much interested on that full push technology. How, as a country, Sri Lanka, we can implement such technology in Sri Lanka?
So I missed the first part of the question. It was a little quiet. So you're asking how to implement such technologies in Sri Lanka?
Yes, we are much rich in biodiversity, and there are a lot of drought tolerance plants in there. So how these technologies can implement in Sri Lanka?
Yeah, so the best way to implement these technologies in Sri Lanka is through collaborations with universities outside of the country. And to be honest, that's what I do in my lab. When I see a really cool application or an advanced technology that I'm interested in bringing to my lab, one of the easiest ways to bring that technology back to Michigan State or back to Sri Lanka is to start mobilizing our students between labs. And that not only gives an opportunity for scientific exchange, but also for cultural exchange. And it really ensures that, the road is paved. And I think the work that Chaturanga and Denim are doing by building this relationship, it's a two-way street. We're benefiting from it here at Michigan State University. And as soon as the pandemic is lifted, we expect to see many students and many scientists and many professors from Rajarata University at East Lansing at Michigan State University. So that's how you're going to implement it. You've got the classroom technology, you've got the foundations of knowledge. And I think by just getting access to this device, and I believe we've left some of the devices there, you have some of the technologies, and we can continue to send these technologies to you. And I think that will help grow not only the partnership, but the adoption. And really, I guess this is a challenge that I pose to the students in the audience. In 5 years, I want you guys to take this technology and make it 10 times better than it is right now, and own it, and develop companies and develop new applications for the betterment of yourself and your university and your country.
Thank you, Brad. So, some more questions? Well, I guess that's pretty much all. We are running out of time as well for the Q&A session. So thank you so much. I'm pretty sure your talk will be the highlight of this morning. Thank you so much again.
Thanks, Denim. Good to see you.
Thank you, Professor Brad, and also Dr. Dinum Pereira for coordinating that session. Ladies and gentlemen, as we are celebrating the Silver Jubilee of Rajarata University, the International Research Symposium of Rajarata University is hosted this year by Faculty of Technology, the newest faculty of the university. It is my pleasure to invite the Dean of the Faculty of Technology, Professor Buddhika Jayatilaka, to address the gathering.
Oh, very good morning all. Atipujaniya Mahasangharasning Avsarai. The Chancellor of the Rajarata University of Sri Lanka, Most Venerable Italavatnave Jnanatilaka Mahanagatero, and Chancellor of the Rajarata Vice Chancellor of the Rajarata University, Professor Sanjuni Ginigaddara, and all the keynote speakers, and deans of the faculties, and heads of the departments, and all the other distinguished guests. So it's with great pleasure I welcome you all to this inauguration ceremony of the symposium, of the International Symposium of the Rajarata University of Sri Lanka. And yeah, so this year is very special. As Pumi said, this year we are celebrating our 25th anniversary of the university. So, and the Faculty of Technology, the latest addition to the family of Rajarata University, got the privilege of organizing this annual symposium going along with the 25th anniversary. So it's an honor to us. And it was not an easy task, especially because of this pandemic situation. We had to face a lot of challenges. And especially at one point, we were thinking, how can we do this symposium? Are we going to do it physically? Because that's what we have used to do. And are we going to do it virtually? But we have zero experience in conducting these kind of symposiums in virtually. And do we have enough resources to do that? All those kind of questions just hit back at us. But I have to say that our staff, especially the staff of the Faculty of Technology and the other representatives of the other faculties, they got together and somehow they managed to come across of all these challenges. And that's why we are sitting here today in this inauguration ceremony. It's really something we need to appreciate. At the same time, this symposium will go into the history as the first ever virtual international symposium organized by the Rajarata University of Sri Lanka. And at the same time, this time with all these challenges, we managed to introduce some new aspects into this annual research symposium. One thing is this pre-conference webinar series. So it was a huge success. Through that, we managed to attract some of the best researchers in the world through this webinar series. A lot of people were attended, and it was a huge success. And in addition to that, at the same time, most of the papers we have submitted into this conference, they will be reviewed again, and the best papers will be sent into the Elsevier publishers. And at the same time, so this is the 6th time we are organizing this annual symposium, and last 5 occasions, it's the same symposium, the annual symposium of the Rajarata University of Sri Lanka, but we had 5 different names. But from this time, we are proposing to fix this name. So we have given a name for the annual symposium of the Rajarata University, known as International Symposium of Rajarata University. The synonym is ISymRU. So what I propose is to keep this name as a unique name for the symposium of the Rajarata University. So then we will have an identity. So everybody, when they see this name, they will identify this as the symposium of the Rajarata University. So I expect from all the people, all the members of the other faculties to go ahead with this name. So let's keep an identity for our university conference. And at the same time, we develop a website. Similarly, in last 5 years, we had 5 different websites, and some of them were good, and some of them were not up to the standard. And this time, we develop a very good website that also we can keep that for our future use. That means we don't have to waste our money and time to develop new websites. We can use the same website and just do the updating at the same. So it will be there all throughout the year. So if anybody needs any details about these conferences, any time of the year, they should be able to download from these particular websites. So those kind of things we introduced into this conference this year. Yeah, so I hope all of you will get benefit of these things. And of course, I'm not going to talk long because this is not a place to talk, not a place to talk to us. Today is a day that the researchers will speak, and we should listen to them. So I will conclude my talk here, and again I would like to welcome you all very warmly to this 6th annual research symposium of the Rajarata University, and hope you will have a very fruitful conference, and I wish all of you good luck. Thank you.
Thank you very much, sir, for being a great captain for the Faculty of Technology ship. මීළඟට මෙම සමුළුව ආමන්ත්‍රණය කිරීම සඳහා මේ ගෞරවනීය ආරාධනය ශ්‍රී ලංකා රජරට විශ්වවිද්‍යාලයේ කුලපති ශාස්ත්‍රපති පණ්ඩිත ගෞරවාර්හ අවසරයි ඊතල වැටුණුවේ ඥානතිලක ස්වාමින් වහන්සේ ඔබ වහන්සේටයි.
මූලාසනයේ වැඩ සිටින පූජ්‍ය ආචාර්ය ඔලකන්වත්තේ චන්දසිරි අපේ රජරට විශ්වවිද්‍යාලයේ අංශාධිපති භාෂා හා තාක්ෂණ පීඨයේ අංශාධිපති අපේ නායක හාමුදුරන් වහන්සේ, ඒ වගේම රජරට විශ්වවිද්‍යාලයේ උපකුලපතිනිය වන මහාචාර්ය සංජීවනී ගිනිගද්දර මැතිණිය, ඒ වගේම රජරට විශ්වවිද්‍යාලයේ හිටපු කුලපති උපකුලපති ආනන්ද මහාචාර්ය ආනන්ද කරුණාරත්න මැතිතුමනි, මහාචාර්ය බුද්ධික ජයතිලක අපේ පීඨාධිපතිතුමා තාක්ෂණ විද්‍යාපීඨයේ පීඨාධිපතිතුමා ඇතුළු මේ අවස්ථාවට සහභාගී වන පීඨාධිපතිතුමන්ලා අංශාධිපතිතුමන්ලා ඇතුළු අධ්‍යයන අනධ්‍යයන සියලුම මහත්තුරුනි, මේ තාක්ෂණ සමුළුව රටට ලෝකයට 21 වෙනි සියවසට මුහුණ දෙන ජනතාවට අලුත් පරම්පරාවක්, අලුතෙන් හිතන පරම්පරාවක් ඉන්න ලෝකයකට ආමන්ත්‍රණය කරන හොඳ අවස්ථාවක් බවට පත් වේ. එනිසා අද දවසේ මෙවැනි සමුළු විශාල ප්‍රමාණයක් ප්‍රායෝගික කරගන්නට පුළුවන්කම ලැබෙනවා නම් ඊටාම වැදගත් කියලා අපි අදහස් කරනවා. ඒ වගේම තමයි බුදුරජාණන් වහන්සේ ආර්ය පරේෂණයේදී නිර්වාණය අරමුණු කරගෙන ඒ ගමන යන හැටිත් ලෞකික භෞතික ලෝකයේ සම්පත් පරිභෝජනය කරලා එයින් මිනිසාගේ සත්‍යයට ස්වභාව ධර්මය සමග ජීවත් වෙන්නේ කෙසේදයයි උන්වහන්සේ දක්වලා තියෙනවා. පැලකට යම්කිසි පැලයකට වතුර දාපුවාම ඒ පැලෙන් ජනේලි දොරවල් උළුවව් පරාල ඔක්සිජන් ඖෂධ වාගේ දේවල් ලැබෙනවා. මේ දරුවොත් එහෙම තමයි. මේ දරුවන්ටත් අපි ගුරුවරු හැටියට ගුරුවරු දෙවිවරු හැටියට සලකන්නේ ගුරුවරු දෙන ශක්තිය දරුවාගේ බුද්ධිය සමාජානුගත කරන්නට, වෙනස් විදිහට ලෝකෙ දිහා බලන්නට, නව නිර්මාණ නව නිපයුම් ඇති කරන්නට දරුවාට හොඳ ආත්ම ශක්තියක්, ජවයක්, බලයක්, ධෛර්යයක් ලැබෙනවා මේ ගුරුවරුන්ගේ ආශිර්වාදයේ සහ ඔවුන්ගේ මඟ පෙන්වීම මෙවැනි සමුළුවන් තුලින්. ඒ නිසා අපිට හුඟක් අවශ්‍ය වන්නේ අපේ රජරට විශ්වවිද්‍යාලය මේ කැලේ කොලේ ඇලේ දොලේ තියෙන මේ වන්නි ප්‍රදේශයේ තියෙන ලොකුම ආභරණය ලෙස අපි සලකනවා. මේ ජීවත් වෙන අහිංසක මිනිසුන්ගේ දරුවන්ට මහා සම්පතක් බවට රජරට විශ්වවිද්‍යාලය පත් කරගන්නට අපි සියලු දෙනාම එකමුතුව මේ රජරට ජීවත් වෙන මිනිසුන්ගේ අවශ්‍යතාවයන් වලට අනුව තියෙන ගැටලු වලට විසඳුම් මේ පරීක්ෂණ තුලින් හොයාගෙන විසඳුම් ලබා දෙනවා නම් හොඳයි කියලා මම අදහස් කරනවා. පළවෙනි එක තමයි වකුගඩු රෝගයෙන් විතරක් උතුරු මැද පළාතේ 21000ක් ඉන්නවා. පිළිකා රෝගයෙන් විතරක් ඉන්නවා 37000ක් විතර. අනුරාධපුර ළමා රෝග ඒකකයට විතරක් 1500ක් විතර වසරකට හෘදාබාධ තියෙන දරුවන් පමණක් පැමිණෙනවා. ඒ වගේම මන්ද පෝෂණයෙන් පෙළෙනවා 5% විතර උතුරු මැද පළාතේ දරුවෝ. ඒ නිසා අපි විශ්වවිද්‍යාලයක් හැටියට මේ මහ පොළොවේ ජීවත් වෙන මිනිස්සු හැටියට අපි හැම කෙනෙක්ම දක්ෂයා, ව්‍යක්තයා, බහුසුතයා, පරේක්ෂකයා හොයාගෙන ඔහුට ශක්තියක් වෙලා එකමුතුව මේ ගමන යන්නට පුළුවන් නම් ඊටාම වැදගත් වෙන. ඒකට තරාතිරමක් ඕනේ. ඒකට වත්පොහසත්කමක් ඕනේ. ඔහුගේ අවශ්‍යතාවය දැකලා ඒකට ශක්තියක් වුණොත් මා හිතනවා ඒ ගමන යන්නට පුළුවන්කම ලැබෙයි කියලා. ලෝකේ දියුණු වෙලා තියෙන්නේ අනාගතයේ ඉඳලා වර්තමානයේ බලාපු නිසාත් වෙනස් විදිහට ලෝකෙ දිහා බලාපු අය නිසායි. අයිසෙක් නිව්ටන් ඇපල් ගෙඩියක් උඩ දාලා ගුරුත්වාකර්ෂණය හොයාගත්තා. අපිට තල් ගෙඩියක් පොල් ගෙඩියක් ඔළුවට දාගත්තත් මේක හොයාගන්න බැරි වෙන්නේ හේතුව තමයි අපි වෙනස් විදිහට ඒ ගැන හිතන්නේ නැති නිසා. පතොක් ගහ කඳු සිකරවල පැලවෙලා කවදාවත් වතුර ඉල්ලුවේ නැහැ. හැබැයි ආ උත්සාහයෙන්ම කොහොම හරි එකේ මල් හට අරගෙන පතොක් වලට ශක්තියක් වුණා. ඒ නිසා අපිට තියෙන භෞතික සම්පත් පරිභෝජනය කරලා අපි නැගිට්ටොත් අපිට නැගිටින්න පුළුවන් ශක්තියක් තියෙනවා. හරියට මේ අපේ ජීවිත බොක්සිං කාර්යයක් වගේ. බොක්සිං කාර්යය කවදාවත් ගුටි කෑවට පරදින්නේ නැහැ. ඇහැ අත පය නැති වුණාට පරදින්නේ නැහැ. ඔහු පරදින්නේ පරාජය පිළිගත්තොත් පමණයි. ඒ වගේ අපටත් අභියෝග, කරදර, ප්‍රශ්න, ගැටලු එන්නේ නව නිර්මාණයක් කරගන්නටයි. ඒ නිසා මහාමාරියා රෝගේ හැදිලා මිලියන 2000ක් මිනිස්සු මැරුණා. ඒ කාලේ හිටපු අයගේ දක්ෂකම නිසා ඒකෙන් ජනතාව ගලව ගත්තා. ඉබෝලා වසංගතේ හැදිලා මිනිස්සු මිලියන 1500ක් විතර මරණයටපත් වුණා. බ්ලැක් ෆංගස් රෝගේ හැදිලා මිනිස්සු 1500ක් විතර මරණයටපත් වුණා. ඒ වගේම චයිනීස් වුණ, හොංකොං වුණ, ඒ වගේම වසූරිය, ඊළඟට මේ සත්තුන්ගේ හැදිච්ච උණ වර්ග මේවා දිහා බලපුවාම ඊළඟට ලංකාවේ දරුණුතුම රෝගයක් තිබුණා මේ රක්තාක්ෂි යක්ෂය කියලා. මේ වගේ රෝග හැදුණා. මේවට ඔක්කොටම මේ විශ්වවිද්‍යාල වගේ පරීක්ෂණ ආයතනවල ජීවත් වෙච්ච බොහෝ අය ඒවට විසඳුම් දුන්නු නිසා අද අපි සතුටින් ඉන්නවා. අද අපි නිරෝගීමත්ව ඉන්නවා. ඒ නිසා අපි හෙට දවසේ ඉපිදෙන දරුවට පවා අලුත් දෙයක් අලුත් විදිහට යහපත් විදිහට නිර්මාණාත්මකව ලෝකයට මුහුණ දෙන්නට පුළුවන්. ලෝකයේ අවශ්‍යතාවයට නිර්මාපකයන් නිර්මාණය කරන්නට පුළුවන් භූමියක් බවට අපි රජරට විශ්වවිද්‍යාලයේ ඇති කරගත්තොත් මා හිතනවා ඒක අප ලැබපු ජයග්‍රහණයක් හැටියට. වෙනදාට වැඩිය 21 වෙනි සියවසට මුහුණ දෙන්නකොට මේ නිව් ජෙනරේෂන් එක නැත්තම් අලුත් සමාජය තුළ දිහා බලනකොට අපි ධර්මයේ අනුවත් ස්වභාව ධර්මයේ අනුවත් මිනිසු නිර්මාණය කරන්න ඕනේ. ඒක තමයි ජීවිතේ මා හිතන්නේ අලුත් තාක්ෂණයට දී යුතු හොඳ දෙයක්. ඉස්සර අපි ඉංජිනේරු මහත්තයෙක් නිර්මාණය කළා, දොස්තර මහත්තයෙක් නිර්මාණය කළා. දැන් අපි ඉංජිනේරුවෙක් නිෂ්පාදනය කරනවා. අද දොස්තර මහත්තයෙක් නිෂ්පාදනය කරනවා. නමුත් ඔවුන්ගේ ಗುಣ නැන බෙලෙන් කිව්වේ ಗುಣ කිව්වේ හෘද සාක්ෂියට, නැන කිව්වේ මොළයට. මේ දෙක තියාගෙන තමයි අපි හදන්න ඕනේ. අපි දැකලා තියෙනවා සමහර වෙලාවල් අවස්ථාවලදී ಗುಣ නැතුව ඥානය විතරක් නැත්තම් නැන විතරක් තියෙන සමාජයක් අද රටේ බිහිවිගෙන යනවා. ඒකට හේතුව තමයි ගොඩක් මේ අලුත් තාක්ෂණය පාවිච්චි කිරීමේදී ජීවගතිය මිනිස්සු තුළ නැතිවීම. ආටිෆිෂල් ඉන්ටෙලිජන්ට් එක යූස් කරන නිසා ඔවුන්ට හොඳ නරක තේරුම් ගැනීමට අපහසුකමක් තියෙනවා ලෝකෙ දිහා බලපුවම. ගැටුම් වැඩි වෙනවා. ඉතින් ඒ නිසා මේ වෙලාවේ මෙවැනි සමුළුවක් ආරම්භ කිරීම ගැන විශේෂයෙන් අපි මේ පහුගිය දවස්වල මහාචාර්ය දිසානායක මැතිණිය කොළඹ විශ්වවිද්‍යාලයේ ළමුන් 200ක් විතර අරගෙන කරපු පරීක්ෂණයකදී හොයාගත්තා මේ ජානවල පිළිවෙල වෙනස් කරන්න පුළුවන් කියන එක, ජෙනෙටික් කෝඩ් කියන එක වෙනස් කරගන්න පුළුවන් කියලා. දැන් මේ මනස තමයි මනෝපුබ්බංග මා ධම්ම, මේ මනස තමයි වැදගත් වෙන්නේ. අපිට වෙනම අභිධර්ම පිටකය කියලා වෙනම තියෙනවා මේ මනස සම්බන්ධ ඒවා. අපේ දැන් අපි හුඟක් දේවල් දිහා කල්පනා කරනවා නම් බලනකොට මේ ඔක්කොම අපේ ත්‍රිපිටකයේ තියෙනවා මේ සියලු දේවල්. ඒ කියන්නේ දැන් අපි ගත්තොත් එහෙම ඥාන බීජවල ශක්තිය පවා ක්ලෝරීකරණයට ආවා. අපි ඇබකස් එකට ආවා. ඊට පස්සේ සයිඩ් රූල් එකට ආවා. මේන් ෆ්‍රේම් එකට ආවා. ඊට පස්සේ අපි දිගටම මෙහෙම ආවට පස්සේ අපි මිනි කම්පියුටරයට ආවා. අවසානයේට අපි ක්ලෝරීකරණය එනකම්ම අපි දියුණු වෙලා ආවා. හැබැයි මිනිස්සෙක් නැතුවයි මේ දියුණු වෙලා ඇවිල්ලා තියෙන්නේ. ඒ නිසා අපි මනුස්සත්වයේ ආධ්‍යාත්මයේ එක්ක මේ ගමන ගියොත් හුඟක් හොඳයි කියලා හිතෙනවා. ඊළඟට තමයි අපේ රටේ පළාතේ විශ්වවිද්‍යාලවල 65% ලෝකේ ගත්තහම පරේෂණ කරලා තියෙන්නේ, නව නිපයුම් හොයාගෙන තියෙන්නේ, නව තාක්ෂණය හොයාගෙන තියෙන්නේ. තාක්ෂණය කියන එක සාමාන්‍ය විනාඩියකට සැරයක් වෙනස් වෙනවා ලෝකේ. ලෝකේ හැමදාම තාක්ෂණය වෙනස් වෙනවා වේගෙන්. මේ වෙනස් වෙන ලෝකේ නොවෙනස්ව තියෙන්නේ නැහැ. නමුත් තාක්ෂණය වෙනස් වුණාට අපි ඒ තාක්ෂණයට මුහුණ දෙන්නට පුළුවන්, අභියෝග වලට මුහුණ දෙන්න පුළුවන් පිරිසක් අපේ විශ්වවිද්‍යාලය තුල නිර්මාණය වෙනවා කියන එක අපට ලොකු විශ්වාසයක් තියෙනවා මේ ඉන්න විද්වත් පිරිස දැක්කහම. ඒ නිසා දැන් කැලිෆෝනියා විශ්වවිද්‍යාලය ගත්තත්, ඔක්ස්ෆෝඩ් විශ්වවිද්‍යාලය ගත්තත් සතියකට නව නිපයුම් විශාල ප්‍රමාණයක් එළි දක්වනවා. අපේ විශ්වවිද්‍යාලයත් අපට සම්පතක් බවට පත් කර ගන්න ඕනේ. රජරට පමණක් නෙවෙයි, රටට විතරක් නෙවෙයි, ලෝකෙටම කරන්නට පුළුවන් ඒ අභියෝග ජය ගන්නට පුළුවන් ශක්ති ප්‍රමාණයක් අපි ගාව තියෙනවා. අපි ගාව ඒ ශක්තිය තියෙනවා. ඒ ශක්තිය අපි පෞරුෂයක් දීලා සමාජානුගත කරලා නව ඇත්තටම විශ්වවිද්‍යාලයක් තුලින් පරේෂණ, නිරීක්ෂණ මේවා ඇති කරලා බෙල්ලා මේ පළාතටවත් අඩු ගානේ විසඳුම් දෙන්න පුළුවන් ධෛර්ය සම්පන්න පිරිසක් ඇති කරනවා ඇතයි විශ්වාස කරනවා. නිසා කාලය ගෙවි ගිහින් තියෙන නිසා මේ වෙලාවේ අදිෂ්ඨානයේ ප්‍රබල නම් බාධා කියන්නේ වචන පමණයි. අපිට අදිෂ්ඨානයක් තියෙනවා නම් අපිට ගමන පුළුවන්. ඒ නිසා ගමන ගැන හිතන්න එපා මහත්තුරුනි, අපි ගමනාන්තය ගැන හිතමු. ඒ නිසා අපේ ගමනාන්තය තමයි ජීවිතේ වැදගත් වෙන්නේ. ඒ නිසා මේවා සංවිධානය කරපු මහත්තුරු සියලු දෙනාටත්, මේවට උදව් කර උපකාර කරපු සියලු දෙනාටත්, ජාත්‍යන්තරයේ ඉඳලා මේවා සංවිධානය කරපු සියලු දෙනාටත් අපි ස්තුතිවන්ත වෙනවා. ඒ නිසා රජරට විශ්වවිද්‍යාලයේ ලොකු ආභරණයක්, ලොකු ශක්තියක් මේ තාක්ෂණ විද්‍යාපීඨයේ දියුණු කරලා, තාක්ෂණ විද්‍යාපීඨයේ තුලින් වර්තමාන සමාජයේ දිනෙන් දින දියුණුවට යන සමාජය තුළට අවශ්‍ය වන්නේ තාක්ෂණයයි. ඒ තාක්ෂණය අපේ විශ්වවිද්‍යාලයේ රටට, ලෝකයට, රාජ්‍යයට බෙදා හරින ආයතනයක් බවට පත් වේවායි ප්‍රාර්ථනා කරන සියලු දෙනාටම සම්මා සම්බුදු සරණයි.
වහන්සේට අපගේ නමස්කාර පූර්වක ප්‍රණාමය ස්වාමීන් වහන්සේ. We are truly honored to have the presence of our newly appointed Vice Chancellor at this ceremony, Professor Sanjeevani Ginigaddara. Madam, I would like to have the honor of inviting you to the lectern to address the gathering.
New knowledge is the most valuable commodity on earth. The more truth we have to work with, the richer we become. ගෞරවහාර අවසරයි ඊතල් රත්නාවේ කුලපති ස්වාමීන් වහන්සේ, සමාජය හා මානව විද්‍යා පීඨයේ භාෂා අධ්‍යනාංශයේ අංශාධිපති Acharya Olaganwate Chansri Swaminu Hansa. Former Vice Chancellor of Rajarata University of Sri Lanka, Professor Ananda Karnaratne, Deans of all faculties, Registrar, Bursar and Librarian, all heads of the departments, professors, academics, other distinguished guests, ladies and gentlemen. Conducting research and dissemination of found out knowledge is one of the biggest responsibilities of any university. Rajarata University occupies a very good research culture. Academics and students of the university are conducting researches in various disciplines. Many high quality researches are going on at the moment in all 6 faculties as per my knowledge. dissemination of that found out knowledge from these researchers to the community is equally important. Today's event has been organized for that purpose. As we all know, Rajarata University is celebrating its 25th anniversary in year 2021. In parallel, this International Research Symposium, ISymRU, has been organized and conducting today. Among the 6 faculties of the university, 1 faculty shoulders this international event every year. This time, the youngest faculty of the university, the Faculty of Technology, is shouldering this event in their capacity of organizing and conducting today. So that is basically with the leadership of Professor Buddhika Jayatilaka, the Dean of the Faculty, and Symposium Chair, Dr. Taturanga Bamunwarachi, as well as the Symposium Coordinator, Dr. Puvudu Kumarage. These leaders, as well as their teams, many thanks for your hard work. Due to prevailing situation, the symposium would be on virtual platform, and let's wish to have a physical international symposium next year if this COVID pandemic dilates. I'm delighted to mention today in this forum that 2 of our researchers from the Faculty of Medicine and Allied Sciences have been listed among the world's top 2% scientists as per the Stanford Varsity's list. This is because of their utmost enthusiasm, commitment, as well as the talents of conducting researches and publishing them in index and high quality journals. But we have high capacity, and we have a lot of intellectuals with very big capacity in other faculties as well. As we all know, as academics, we might be different. Sometimes, some academics may be very good at their research, conducting research and publishing them and writing, so they might be really good at them. Some academics, they may be good at administrative work. Meantime, some academics may be good at both, balancing administration as well as research. My invitation is the first and third category. Please recognize your fullest capacity and capitalize that for conducting very good research and publishing them in high quality journals. However, I have a big worry that a big percentage of this research and the found out knowledge is ultimately ending up with the publication and publications. There is no exception in Rajarata University as well. A minute number, very rarely some of the researchers are going to the general public in the commercialization endeavors. And the research outputs are rarely utilized by the general public. We know that the present government's higher education vision is the government universities are to be centers for economic development, agents of innovation, and incubators of entrepreneurship. In parallel to this vision, we have to jump into new focus. I feel this is the high time and right time for that. So let's focus on our attention when we are again writing research proposals and planning for research and conducting research and development activities. We'll focus on research for the commercialization endeavors. Rajarata University has already focused and established a cell to help such research outputs or let's say innovations or inventions to bring to the mass market for the benefit of general public. So my special request in this forum and to the university community is, please focus on your future research to be ended up with innovations and inventions leading commercializations as well. This definitely brings the Rajarata University name to the community. Again, thank you very much, our newbie, the Faculty of Technology, for organizing this wonderful session today and tomorrow, and including this inaugural session. And thank you very much, the keynote speaker, Professor Brad Day, for accepting our invitation to deliver this keynote speech in this bright morning. And I wish the rest of the symposium work as well as technical sessions to be a success. Thank you very much.
Thank you very much for that insightful and inspirational words, madam. Coming together is a beginning, and keeping together is progress, and working together is a success. The International Research Symposium of Rajarata University 2021 is such a team effort that we are humbly proud of. We are also blessed with many helping hands. I would like to invite Dr. Mithila Bandara, the Secretary of ISymRU 2021, to propose a vote of thanks to these helping hands.
Chancellor of the Rajarata University of Sri Lanka, the most venerable Italavatnu Eve Gnanatilaka Thero, and the Vice Chancellor Professor Sanjeevani Ginigathara, all the deans of the faculties, invitees, colleagues, and dear students. It is my privilege to have been asked to propose a vote of thanks on this special occasion. Today, we have marked the beginning of the 6th International Symposium of Rajarata University, together with the participation of the national and international research community. We believe that this will be one of the proudest days in the history of the Rajarata University of Sri Lanka. But I must say here that organizing an international symposium is a challenging task. This wouldn't be possible without greater preparation, wise working hands, support from every corner, and the blessings. So now it is our time to convey our gratitude to all those who have extended their support to make this event a greater success. At the outset, on behalf of the organizing committee, I would like to express my profound gratitude to the Chancellor of the Rajarata University of Sri Lanka, the most venerable Italavatnu Eve Gnanatilaka Thero, for his blessings and motivation. Thank you for your participation in this inaugural session. We also humbly thankful to our newly appointed Vice Chancellor, Professor Ginigathara, for adjusting your busy schedule and being with us here today. And also, I would like to take this opportunity to congratulate on your new appointment. Congratulations, madam. Next, I'm happy to offer my sincere thanks to the Dean of the Faculty of Technology, Professor Buddhika Jayatilaka, for active guidance and support. And thank you for giving your valuable time in fixing many things related to the symposium. Our words may not be capable to express our sincere sense of gratitude to the keynote speakers, webinar speakers, session chairs, and the judges. Thank you for accepting our invitation and graciously agreeing to share your knowledge. We hope your presence will enhance the experience of all the participants. I also take this opportunity to thank all the reviewers for providing your valuable comments and helping towards the improvement of the quality of the papers presented in the symposium. I would like to thank all the authors who have submitted their valuable research to the symposium. We hope the symposium will be an outstanding opportunity for you all for networking and personal development. Next, I convey my sincere thanks to the sponsors of ISymRU 2021. Especially, my thanks goes to University Research and Program Committee, Asian Development Bank, and all the other sponsors for funding and providing and supporting the event to bring this symposium to this level. And equally, on personal behalf, I am thankful to all the faculty coordinates and the organizing committee for their dedication and support to make this event a greater success. And also, I would like to thank Dr. Ravindra Jayatilaka for the nice floral work that you see here today. My special thanks goes to Dr. Chaturanga Bamunuarachige, the symposium chair, and the symposium coordinator, Dr. Pubudu Kumarage, for putting their last complete week in organizing this symposium. As a faculty, the Faculty of Technology is pleased to be able to hold this 6th International Symposium. So, though in last, most importantly, I would like to thank all the academic, non-academic, and the students of the Faculty of Technology who volunteered and worked hard selflessly for the success of the event. At last, I would like to thank you all for being here, for coming and being here in this morning. And I wish you all the very best and hope you all will have a productive symposium. Thank you.
Thank you. Ladies and gentlemen, let us all rise for the national anthem.
Sri Lanka Mata, Upper Sri Lanka, Namo, 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 Namo Mata. Sundara Sri Karini,
With that note, we are marking the end of this inauguration ceremony. Ladies and gentlemen, refreshments are ready at the lobby. May you have a wonderful day ahead and a fruitful morning.